Okay. Hello, ladies and gentlemen. These are your notes on cell regulation. And I hope that going through this helps you understand the cell cycle just a little bit more. The key questions that we are trying to understand today is how is the cell cycle regulated and how are cancer cells different from regular and or healthy cells? So let's start with the first idea, this whole idea of what it means to have cell regulation. Any kind of control on cell division is going to be cell regulation. When cells come into contact with each other, we know that they usually stop dividing. When an injury happens, we know that cells at the edges of the injury will start dividing very, very rapidly to start the healing process. But the question is, how do they know to do this? How do they know when to stop? How do they know when to start up again? And the answer is regulatory proteins. Regulatory proteins basically are going to tell the cell when it is time to replicate it. And also, kind of flip side of that is when it's time to stop as well. There are many signals or many regulatory proteins that help control the cell cycle. Scientists used to believe it was one protein, and what we have found out is that it's many, many proteins. So basically, the cell cycle, remember we talked about that already, interphase, which is G1, S, G2, and then the M phase, which is made up of mitosis and cytokinesis, is controlled by regulatory proteins, and those regulatory proteins can exist both inside and outside of the cell. So let's take a look first at our internal regulators. These are regulatory proteins that respond to internal conditions inside of the cell. These are our checkpoint proteins. So remember, we talked about checkpoints last week. There are three main checkpoints during cell division. There is the G1 checkpoint. The G1 checkpoint is going to check for, is there enough nutrition? Is there enough growth factor? Is there any DNA damage? If everything checks out and everything's good, then it allows the cell to continue on to the S phase. If everything does not check out, then that cell may enter into G0, which is a resting stage. So it's going to stop the process of the cell cycle. We have G2 checkpoint. The G2 checkpoint checks for cell size, and it looks at the DNA replication. Is the cell the correct size to start replicating? Are there any problems in the DNA? If so, then we'll go through apoptosis. We'll explain that in a minute. If everything checks out, if everything's good, it moves on into the M phase, right? M phase is the mitotic stage or the mitosis stage, which is mitosis and cytokinesis. Mitosis being the division of the nuclear property of the nucleus, and cytokinesis being the division of these cytoplasm. In metaphase, there is another checkpoint, and it's going to check that did the chromosomes attach the spindle correctly. Remember, in metaphase, everything lines up down the center, and those spindles get connected, right? And we know that next is going to be anaphase. We're going to separate those sister chromatids. If there's any problems with the spindle attachment, that could cause DNA problems later on in the process. So again, we're checking for that. And again, these internal regulators are checking these. So what are these actual internal regulators? They're called cyclins. These are groups of proteins that regulate the cell cycle. They regulate the timing that the cell cycle happens in by rising and falling concentrations of cyclins. So we can see here in this graph that and this is just, I think it's an oyster cell. Cyclin increases, and then starts to go down. And then it goes up, and then it goes down again. And as time goes on, we can see these cyclin levels increasing and decreasing as we go through G1, G2, G1, S, G2, and the M phase of the cell cycle. All right, oops, my bad. Cyclins, which again, internal regulators, there are different types of cyclins that control different checkpoints. So we can see that cyclin E is controlling the G1 phase. Cyclin A controls the G2 checkpoint, and then cyclin B controls the metaphase checkpoint. And again, we know that what they're controlling based on the amount of concentration present at what time during the cell cycle. Cyclin D, you can see kind of goes rises and falls throughout the entire process. This is just the one that's telling the cell, hey, it is time to go through the cell cycle. Now, remember how we talked about, well, sometimes they fail these checkpoints. What if the DNA didn't replicate properly? What happens if the cell isn't large enough? What happens if the spindles did not attach to those chromosomes correctly? If it fails any of these checkpoints, that cell may be signaled to go through apoptosis. Apoptosis is a series of steps that causes the cell to self-destruct. So if you've ever watched any kind of sci-fi movies and the captain of the ship's like, we're going to self-destruct the ship, that's kind of what happens in apoptosis. Because there is damage, because there is a error in the cell, and you don't want that cell error to be replicated, the cell is instructed to self-destruct. Okay? There are external regulators. Again, cyclins are the internal regulators, but there are also external regulators to the cell cycle. Any external regulators is any protein that responds to conditions outside of the cell. These are the ones that are going to tell our cell cycle to either speed up or slow down, okay? An important group of external regulators are known as the growth factor regulators, and these stimulate growth and the division of cells. Growth factors are really super important to embryo development and wound healing. Again, growth and repair. There's no time in the development of an organism where the cells are dividing faster than when you are in the embryonic stage. In other words, when you are basically inside the egg still, or maybe you're the puppy inside the mom dog's womb. When you are in that stage of development, you are developing very, very quickly, which means growth factors are really important. Wound healing, we've already talked about ad nauseum. There are other external factors, regulators that will help slow down growth. And these are the ones that keep the tissues from disrupting each other. Remember, we don't want our heart growing into our lungs. We don't want the stomach growing into the spleen. We don't want our liver growing into our intestines. That would be a mess. And these external regulators can be used to slow and stop growth when the tissues, to keep the tissues from disrupting one another. So the question becomes, what happens if this fails? If we have uncontrolled cell growth, that is a disorder which we call cancer. Cancer is when our body loses the ability to regulate cell growth. So cancer cells do not respond to internal and or external regulatory signals. As a result, the cell cycle is disrupted and the cells grow in an uncontrolled manner. These uncontrolled growths are called tumors. Tumors do not function as healthy tissue. Tumors invade the surrounding healthy tissue. Tumors will absorb nutrients and block nerves. And when they do these things, they prevent your organs from functioning properly. What is the problem with that? Well, if your tissues or organs are not functioning properly, then it's going to throw off your homeostasis, which can be fatal. We have talked about homeostasis before. This idea of maintaining a nice, constant internal environment. If my tissues and organs, which help to maintain homeostasis, are not working, I cannot maintain homeostasis, which means that it could be fatal to the organism. Okay, that about wraps up these notes, ladies and gentlemen. Hope that helps. Have a great day.